So cytosol is not just a goo. Cytosol is actually organized pretty well, and the cellular structure is supported by cytoskeleton. So cytoskeleton makes sure that the cell is well organized, that it's well shaped, it suspends organelles, it moves organelles, it enables the movement inside the cell. Different parts of the cytoskeleton change, reform, break down, and that allows cells to move and to change shape itself. There are three types of cytoskeleton structures. Microfilaments, shown here in the center, consist of a protein actin, and they are necessary for movement, contractions of the muscles, establish the shape of the cell, and they can be formed and destroyed and formed back. Intermediate filaments, shown on the right, consist of a protein keratin. They're like cables, okay? So they establish the basic cellular structure. They're very stable. They rarely are destroyed. Basically, if they're destroyed, the cell is practically gone. Microtubules, formed of the protein called tubulin, and they're necessary for the movement of the chromosome during the cell division, and they also are essential for the movement of the organelles. External appendages of the eukaryotic cell include flagella and cilia. Here on the bottom, you can see cilia. Cilia, a short extension that look like eyelashes, and these line respiratory tract, they move mucus, they line fallopian tubes. Basically, the main function of cilia is to move mucus, move things that are outside of the cell. Flagella are whip-like structures. That's the cross-section of flagella. Whip-like structures that move the cell forward. The only human cell that is moved by flagellum is the sperm cell. Now, endoplasmic reticulum is the network of membranes, basically membrane-bound lattice, that is directly connected to the double membrane nucleus. Rough endoplasmic reticulum contains ribosomes that are necessary for the protein synthesis. Ribosomes are fairly small organelles that are composed of proteins and RNA, and the function of ribosomes is the protein synthesis. I want to highlight that while endoplasmic reticulum is membrane-bound structure, ribosomes are non-membrane bound. Unlike rough endoplasmic reticulum, smooth endoplasmic reticulum contains no ribosomes. It's responsible for synthesis of fats and steroid hormones. Both smooth and rough endoplasmic reticulum produce vesicles. Vesicles are essentially the transport structures that move the products of fat synthesis or protein synthesis to wherever they are needed. So these vesicles produced by endoplasmic reticulum either deliver the products of the synthesis to the membrane of the cell for exocytosis, or to the Golgi complex for the further processing and packaging. And the function of Golgi complex, shown here, and this is the micrograph, is to the function of the Golgi complex is to process proteins, modify them, modify fatty acids, okay? It's basically a stack of saccules, stack of structures that look like very, very elongated vesicles. One face of the endoplasmic reticulum of the Golgi complex faces endoplasmic reticulum. Another face faces the plasma membrane. And what happens there is proteins and fatty acids are being modified, changed, and then packaged and transported to different parts of the cell or to the membrane for exocytosis. Lysosomes, as I mentioned, they contain digestive enzymes, okay? Various hydrolytic enzymes, acid. So lysosome fuses with endosome, breaks down the content of the endosome transporting different stuff into the cell. So basically, when your cells break down the content of endosomes, for instance, bacteria, they destroy bacteria by fusing endosomes with a lysosome, and the content, it's like I mentioned before, it's like a stomach, okay, but it's a separate stomach that can fuse with the mouth. So lysosome fuses with the endosome. When they content mix, endosomal digestive proteins, digestive enzymes break down everything. Nucleus. Double membraned organelle, contains all cell genetic information. Here you can see two membranes, one membrane, another membrane. These are nuclear pores, necessary for the transport in and out of the nucleus. Now, pores are pretty small, which prevents DNA from spilling out of the nucleus, but allows the transport of proteins and other essential molecules inside of the nucleus and outside of the nucleus. So again, it's double membraned, its envelope is double membrane, and it contains the pores. Inside of the nucleus, you can find non-membrane bound structures called nucleolus. The function of the nucleolus is to produce ribosomes. Mitochondria, we mentioned that the powerhouse of the cell. Mitochondria produce ATP. Cellular respiration, essentially the process responsible for the ATP synthesis, occurs in the mitochondria. You can see that it's also double membrane. You have it as outer and inner membrane. Inner membrane is folded. These folds are called cristae. And basically mitochondria use oxygen and glucose, and sometimes fats, to produce ATP and releasing carbon dioxide and water. Now we're going to stop at this point, and we're going to discuss cellular respiration later in the class.